So people will do a lot of things to lose weight. Some of them wiser than others. Come on. Do folks have any questions they'd like to ask about the last chapter, about chapter 12 that dealt with oxidative phosphorylation, with the idea of electron transport chain and the mitochondrial inner membrane, creating a proton gradient, using that to make ATP. Any general questions about that before we move on to our next topic? Okay, well, let's get started then on photosynthesis. This is one of the few topics that we're going to talk about that we ourselves do not actually do at all. On the other hand, we live in Iowa, so we have to love photosynthesis. But in fact, anybody anywhere should love photosynthesis because it keeps you alive. In very broad terms, what photosynthesis is going to be doing is capturing carbon dioxide using solar energy to make 2 and 3 carbon intermediates. So this is the place where you actually can start to create some of the things with which all those other pathways can manipulate. In other words, those 2 and 3 carbon intermediates had to come from somewhere. And the somewhere they came from almost entirely is from photosynthesis. So that's pretty important. It's also, though, going to be interesting because it's going to have a lot of overlaps in different ways with either pathways we've already seen or pathways that we're going to be seeing. So we're going to see that there are going to be some similarities to oxidative phosphorylation. We're going to see similarities to gluconeogenesis, not too surprising since the goal of photosynthesis is to make sugar ultimately, and the pentose phosphate pathway. So a lot of the things we're going to see in talking about photosynthesis are not going to be completely new to you. But the plants do little cute things with them that make them unique and special. Broadly speaking, we divide photosynthesis into 2 kinds of reactions. We talk about the light reactions, and they're called the light reactions because they only happen in the light. They're ones that can capture the energy from photons, particles of light, in order to make high energy electrons. Well, we're going to see the electron flow moves protons, so that should sound familiar. We're going to see how the proton gradient makes ATP. Again, it should be familiar. And the really cute trick that makes this unique and makes it possible for us to be alive is that the electrons from water are going to be used to reduce NADP plus to NADPH. And in the process, oxygen is released. There are some kinds of photosynthesis that don't release oxygen, and if people are interested, we'll talk about them too. But the kind that makes the most difference to most of the world is the kind that releases oxygen. So those are light reactions. Those only happen in the light because they're driven by light energy. The second kinds of reactions are often called dark reactions because they could, in principle, happen in the dark. They often happen in the light. But they use the ATP and NADPH that's made by the light reactions in order to fix carbon, in other words, to take carbon dioxide out of the atmosphere, and to reduce it to make sugar molecules. All of this rather complicated chemistry happens in a particular organelle in land plants. We're going to see blue-green algae have many similar things. Let's talk about land plants to begin with. And at first glance, this might kind of look like a mitochondrion, which would not be too misleading, a first impression. There's an outer membrane, and there's an inner membrane, but the interior of the chloroplasts is generally so enriched in inner membranes that they actually have discrete areas within it that are called thylakoids. This is a transmission electron micrograph to show you some of the elaboration of the membranes that's involved. And so the thylakoid membranes are analogous to the inner membrane, so that the interior, the empty space of the thylakoids, which is called the thylakoid lumen, is in mathematical terms or topological terms the same thing as the intermembrane space, the space between the inner mitochondrial membrane and the outer mitochondrial membrane would be in a mitochondrion. So you have membranes inside the chloroplast, and they create a space called the thylakoid lumen. The thylakoids are going to be the place where the light reactions are all going to happen. And this is showing you some of those features, setting you in context a little bit for you. At this level, you can see a leaf, such as you actually see with your own eyes. The blow up here is blowing one individual cell of that up, so you can see an individual plant cell here. You can see that it has a nucleus. You can see it has a vacuole, but it also has a number of these chloroplasts, which just means little green bodies. And this is showing you then the chloroplast diagrammatically again, the outer membrane, the inner membrane, and the thylakoids. And then if we look at the thylakoid membrane in a little sort of molecular diagrammatic detail, what we find is something similar to what we saw in the mitochondrion. We see membrane complexes, multi-protein complexes embedded in the membrane that are going to be involved in electron transport. So there's going to be a photosystem 2, a photosystem 1. Photosystem 2 is going to be one of the light capturing systems, photosystem 1, light capturing, and a variety of electron carriers between these things. These complexes are going to do what we saw in the mitochondrion. They're going to move protons, and they're moving them into the lumen of the thylakoid. So the lumen is analogous to the intermembrane space for the mitochondrion, and we're going to build up a proton gradient there. And then, using that proton gradient, we have again a large two-part complex, an ATP synthase that's called CF1, CF0 for chloroplast F1, chloroplast F0 that's going to make ATP. So that's the grand overview of what we're going to be seeing. But to go further into this, we're going to have to think a little bit about photochemistry, which sounds like an odd combination of things. Many molecules can interact with light one way or another. And the way to think about that chemical interaction or photochemistry is that a photon, any given particle of light, you can think of it as a little packet of light energy. Excuse me. And whether or not any given particular photon can interact with a particular chemical is going to depend on the molecular structure. And the particular details of the structure will determine which photons it can or can't absorb. And when we look at plants, and especially when we look at chloroplasts, we see a number of chemicals that are extremely good at absorbing light. We call them photoreceptors. And this is some examples of what those structures look like. Chlorophyll A here is one you probably have heard the name of before. You can see here a porphyrin ring, which should remind you of heme. Heme is another porphyrin ring. But in the case of chlorophyll, it has magnesium in the center instead of iron. And there's some other little details off the side that give it spinachy bits that have a variety of things. Here is phycocyanin. This is a light collector from a variety of red and blue-green organisms. And we call this a linear tetraperol. You should see that if you sort of mentally fold this around into a ring, you can see it would be quite a bit like a heme ring. And in fact, heme is this thing folded into a ring. Here's a third one. Here's beta-carotene. It's present in a number of the vegetables that your parents tried to get you to eat. And it's a precursor for some other things. A feature you should probably have noticed already just by kind of looking at it is how common these conjugated double bonds are. And so you can see there's a lot of double bonds in these structures. And in many cases, they alternate with single bonds. So these things are kind of molecularly set up to twang like a harp string when they get hit by a photon. And it's in the precise detail of where those bonds and so forth are as to which frequency of light they can absorb. And this gives you a feeling for which frequencies different light photons will be absorbed by which molecules. This is what's called an absorption spectrum. So this is, on this axis is showing how much of it is absorbed. And on this axis, we're looking at how long the wavelength is. Short wavelength is more energetic. It's closer to blue. The longer wavelength is less energetic. It's closer to red. That's why we talk about infrared as being even longer than that and ultraviolet as being even shorter than that. But this is the visible spectrum. The solid red line, solid black line there is the solar irradiance, the light we get in from the sun. And what you'll notice here is that different molecules capture different parts of that spectrum. So here is chlorophyll A with a big peak in the blue. Here's chlorophyll B, sort of near it. Here are the carotenoids kind of straddling it. Phycocyanin quite a bit further down. There's a second peak down here for chlorophyll B to kind of pick up some of that red light. But if you look at those all together, they're capturing a significant fraction of the solar spectrum. So the first thing that has to happen for photosynthesis to start going is the light energy has to be absorbed. And so it's going to be absorbed by some molecule, and then that energy is going to be transferred. And so let's say for the moment that a chlorophyll molecule has absorbed a photon, and it's now an excited chlorophyll. It's literally vibrating. So it has energy in it that it didn't have before. It's an excited molecule. What happens next depends on what happens to the energy in that excited molecule. One possibility is it can just relax back to being chlorophyll and give off that energy as heat. Nothing very exciting there. It can give that energy off as light. It can fluoresce. If you've ever noticed in the fall how the trees glow, they actually glow. They are actually giving off a certain amount of light. We can have transfer that electron so that energy can be transferred to some other compound that's now excited. Or we can have a photooxidation event. So we can have some compound getting reduced, and chlorophyll has been oxidized. And it's this possibility for generating an oxidant or a reducing agent out of chlorophyll that it makes photosynthesis possible. In principle, you'd like to get all the light that comes in, or at least a good fraction of it. And in fact, the proteins and pigments of photosynthetic organisms are organized in order to make that more efficient. This is giving you just a little glimpse of how that can take shape. Here you can see ribbon diagrams of the protein part, and then the colored bits are various chromophores, things that are pigmented or colored. And the proteins are holding those chromophores in an organized way so that they can act as an antenna to capture light. Here you're looking at the side of one of these kinds of arrays. Here we're looking down the center of it. Here we're looking principally at the pigmented parts. And so it acts as a really efficient little tube, like a light tube almost, to capture the light. Now what's interesting is that these different antenna pigments can transfer the energy to a reaction center. So if these are all examples of molecules that can absorb light, this light particle coming in excites this one, but it can transfer that energy from one to another molecule until it gets to the reaction center. And the reaction center is the specific molecule that's set up in just the right way to do the next set of reactions we'll be talking about. So in thinking about photosynthesis as a light-driven electron transport, we're going to talk first about photosystem 2, which might sound funny. Why do we start talking about photosystem 2? Well, it's called photosystem 2 because it was discovered second. It happens as an accident that it evolved second. But it is the first step in the pathway of photosynthesis in land plants, in blue-green algae, and so forth. Photosystem 2 is a light-driven oxidation reduction enzyme. That is, it can use light energy to drive oxidation reduction reactions. The chlorophylls absorb photons either directly or by transfer from some other antenna pigments. They're then going to transfer that energy to a specific reaction center, a specific chlorophyll called P680. And it's called P680 because it has an absorption peak at 680 nanometers. And when that energy is transferred to P680, it becomes excited to P680 star. P680 star is extremely good at giving off electrons. It's a very, very good source of electrons. And when it does that, it becomes P680 plus. So if we look over here, we have P680, the light energy coming in, excites it to P680 star. It gives off that electron. P680 plus it's lost an electron. It's been oxidized. We need to get electrons into that, and the new electrons for that are going to come from water. And so that's part of the amazing thing that photosystem 2 can do, is it can get electrons from water in order to keep this cycle going, so that you're going to have the P680 pigment, when it gets excited, give off its electrons. Now it's oxidized. It needs electrons to be reduced back and do its thing over again. It turns out that the machinery to do this is a very substantial, massive complex. Just as there was complex 1 in the mitochondria, that was the first stage in the electron transport, P680 is the first complex in the electron transport for photosynthesis. And you can actually see it in the membrane. It's big enough to take up, detectable space. Here, this is the thylakoid membrane, and the lumen side is the side that is towards the hollow space of the thylakoid. The stroma is like the meat that fills the chloroplast. It's analogous to the matrix in the mitochondrion. It's got the rest of the stuff. And electrons are going to travel from P680 to other redox groups, just as we saw in the mitochondria, that any one complex had several kind of resting places in it for electrons. And then on the stromal side, the electrons are going to get transferred to plastoquinone. Remember, ubiquinone in mitochondria, chloroplasts have plastoquinone. So it's another hydrophobic electron carrier. And in this reaction, it's going to use up 2 protons from the stroma, and so it's going to start creating a proton gradient. A lot of analogies here to complex 1. This is showing you how some of these electron carriers are actually physically oriented relative to each other. Here is the chlorophyll P680 molecule. And its electrons are going to take sort of stopping points along the way in molecules like phaophytin. Here's a bound plastoquinone. So this is a plastoquinone molecule that is part of the complex on a permanent basis. There's an iron in between there. And then finally, the electrons are transferred to reduce plastoquinone. So a lot of analogies to oxidative phosphorylation. We haven't yet made any oxygen. Photosystem 2 is the system in photosynthesis that generates oxygen. We haven't done that yet, so let's think about that a little bit. This is one way of thinking about the events during photosynthesis where we look at changes in the redox potential. You may remember that when we talked about mitochondria, we talked about being able to compare redox potentials of compounds. Here's P680 by itself. When it absorbs a photon to become P680 star, it's turned into a very good reducing agent. And when it loses that electron and becomes P680 plus, P680 plus is the strongest biological oxidant known. It's an extremely good oxidizing agent. It is such a good oxidizing agent, it can actually steal electrons from water. Similarly, P680 star is a very good reducing agent. It's very good at giving away electrons. It's all psyched up and wants to throw that electron off onto something like phaophytin, plastoquinone A, or plastoquinone. So this is sort of a summary diagram of this is a lot more vivid on my screen than I'm up there. This is a summary diagram of photosystem 2 embedded in the thylakoid membrane. It's going to have be able to absorb energy here and strip electrons. An electron from the chlorophyll will be transferred to make a reduced plastoquinone here. In the process, we're pulling protons out of the stroma. In a separate part of the complex, we have the oxygen evolving machinery that can take electrons from water, generate oxygen gas, and release some protons. So we're releasing protons through the process of getting electrons from water, and we're creating a proton difference in the process of reducing the plastoquinone. Okay. Where does the plastoquinone go? Okay. It's going to freely diffuse in the inner mitochondrial in the thylakoid membrane. and it's going to encounter another complex called cytochrome B6F, another membrane complex. And here we have reduced plastoquinone. It's going to be moved through a variety of electron carriers within that complex and ultimately transferred to a peripheral membrane protein called plastocyanin. Here is a ribbon diagram of plastocyanin. Plastocyanin has copper. You might be reminded a little bit of cytochrome C. This is playing an analogous role to cytochrome C, but it's using copper to do it, and so it's green instead of red. And you can see here plastocyanin in the copper 2+ state going down to the 1+ state. It has been reduced. It has received electrons. And this is giving you a sense of the sort of molecular landscape inside the cytochrome B6F complex. And it actually has a number of structural resemblances to cytochrome BC1 in the mitochondrion. In addition to irons in there, almost all cytochromes are going to have heme irons. There are also sulfur iron centers in there. And you can guess by seeing this alluded to up here that there's another Q cycle going on here. That just as in the mitochondrion we had to use the Q cycle to accommodate the fact that cytochrome C only takes one electron at a time. Plastocyanin also takes only one electron at a time, and there's a Q cycle associated with cytochrome B6F for the same reason. And again, we're seeing protons moving here from the stroma into the lumen of the thylakoid lumen. Okay. Next stop in the electron transport sequence is photosystem 1. It was discovered first. It evolved first. It is, like photosystem 2, a light-driven oxidation reduction enzyme. It can capture light and catalyze redox reactions. Just like photosystem 1, there will be chlorophylls that can absorb photons. They can transfer that energy to specific molecules that are called reaction centers. Slightly different reaction center, P700. So photosystem 1 has a slightly different absorption spectrum than photosystem 2. When that energy is transferred to P700, it becomes excited to P700 star. P700 star also likes to give off electrons. And so it's going to give off its electrons to something else, and in the process become oxidized to P700+. We need somehow or other to restore P700+. In this case, those electrons are coming from plastocyanin. So this is that diagram again. P700 gets excited to P700 star. It can give off its electrons. The electrons that it's passing off, this is showing you sort of the structural components, and again, they look somewhat like the ones in photosystem 2, but the ultimate recipient of the electrons from P700 star is going to be ferredoxin. Ferredoxin gets its name because it has iron in it, ferra, and redoxin. So ferredoxin is an electron carrier that is an iron-rich molecule. This is the structure of ferredoxin. There's the iron in there. Ferredoxin is in the stroma of the chloroplast. It's on the side away from the lumen where the protons are accumulating. That positively charged P700+, we need to restore it if we're going to keep the cycle going. It's going to get its electrons from plastocyanin, and plastocyanin will be oxidized. P700 is reduced back to P700, and it's ready to go again. So some similarities to the structure of photosystem 1, differences in who's giving the electrons and where they're getting them from. So what are we going to do with those electrons from ferredoxin? Well, we can use electrons from ferredoxin in a couple of ways. One way is we can use ferredoxin. So here we have some reduced ferredoxin. We can use those electrons to make NADPH. So that is one of the ways in which ferredoxin gets used, is to make NADPH. There's another way, though, in which ferredoxin gets used, and that's in something called this is the one that's called non-cyclic electron flow. If the electrons from ferredoxin are transferred to NADPH, to make NADPH, the electrons originally came from water. So they're starting from water and doing stuff and ending up in NADPH. Photosystem 1 itself does not pump protons. It does not pump protons. It just has this ability to reduce ferredoxin. The whole combination of photosystem 2, cytochrome B6F, and photosystem 1 is called the Z scheme. It's a relatively sort of new one, meaning only 3 billion years old, because it's something that the blue-green algae came up with, and it looks like this. So when you have non-cyclic electron flow, you have two photo events, one in photosystem 1 and one in photosystem 2. In the first photo event, P680 is excited to P680 star, and there's going to be an associated stripping of electrons from water to restore that. So the electrons in this molecule came ultimately from water. P680 star is energetic enough to transfer those electrons to plastoquinone, which can transfer its electrons to cytochrome B6F, which can transfer it to plastocyanin, a lipid-soluble carrier, a water-soluble carrier. The second photo event in photosystem 1 is going to generate P700 star and need the electrons from plastocyanin to restore that. And those electrons can get transferred to ferredoxin, to NADP+ to make NADPH. This is called the Z scheme because there are these two jumps in redox potential. Those two jumps are both driven by the photo events. And then electron transfers from those excited states. And in that scheme, electrons have to be supplied by water. There's another way of using the electrons on ferredoxin, though, and this is something called cyclic electron flow. And in cyclic electron flow, it's much simpler. It looks like this. Photosystem 1 gets excited. It makes reduced ferredoxin. Reduced ferredoxin can transfer its electrons to cytochrome B6F. Cytochrome B6F can transfer them again to plastocyanin, and around and around. The nice trick there is that the cytochrome B6F can pump protons. So in cyclic electron flow, you have some proton pumping done by cytochrome B6F, and the reaction overall is driven by light absorption in photosystem 1. This is the oldest form of photosynthesis. It predates even blue-green algae. There are things called sulfur bacteria and purple bacteria that can use light energy, and they deposit sulfur rather than giving off oxygen gas. Very, very old process. If we were to compare cyclic electron flow to that previous diagram, it would look something like this. The light event can excite P700. The electrons can go to ferredoxin, but now they go from ferredoxin back to cytochrome B6F, plastocyanin and around. And cytochrome B6F is pumping protons, so we can make ATP, but we cannot make any net NADPH. Right? The electrons are just going around and around and around like that. Okay? So it provides ATP, but it does not make available new reducing power. So far, we've had light events in photosystem 1 and photosystem 2, and we've had pumping events in photosystem 1, cytochrome B6F. So we're generating a proton gradient, which should sound very familiar. This allows chloroplasts and blue-green algae to do what's called photophosphorylation. We've had substrate-level phosphorylation with pyruvate kinase and in the citric acid cycle where we make ATP or GTP from intermediates. We've had oxidative phosphorylation. It was driven by transferring electrons to oxygen. This is photophosphorylation. This is making ATP driven by light energy. So the chloroplast can use that proton gradient to drive ATP synthesis. And in fact, it looks a great deal like the events that happened in the mitochondrion. Similar lollipop-type structure. It's again in the membrane, so you can see that the ATP synthase is in the thylakoid membrane. It again is oriented with a seed F0, the chloroplast F0 embedded entirely in the membrane, and the CF1, chloroplast F1, sticking out of it. And again, the CF0 is going to allow for protons to flow from the high end of the gradient in the lumen through it into the stroma. It's going to spin the CF0 in circles, and that spinning is going to drive ATP synthesis on the CF1 component. So it's going to use that change in conformation of the spindle to drive ATP synthesis. If we look at the linear electron flow, using both photosystems, starting with electrons from water and ending up with NADPH at the other end, we can do some bookkeeping on that. 8 photons, so 8 particles of light that are absorbed, will pump 4 protons at photosystem 2. They'll pump another 8 protons at cytochrome B6F, and that overall will release 1 oxygen gas molecule. That would be a total of 12 protons, and the exact equivalence of how many protons to how many ATPs can vary depending on the exact structure of the CF0 subunit. Any questions so far? Everything that we've talked about so far is carried under the term light reactions. They're reactions that will only happen in the light because they're driven through photosystem 1 and photosystem 2. You have to get that excited chlorophyll for anything else to happen. The other big part of what goes on in photosynthesis, besides this light-driven redox system, is carbon fixation. And carbon fixation is the process by which plants and algae can take carbon dioxide gas molecules and fix them into some form of useful carbohydrate, turn them into organic carbon instead of inorganic carbon. The carbon fixation reactions are sometimes called the dark reactions because they're using the products of the light reactions. They will need to have both ATP and NADPH in order to occur. They don't necessarily happen in the dark. They may or they may not, depending on the plant's physiology, but they could, in principle, if you supplied the ATP and the NADPH. The actual event of carbon fixation is going to be accomplished by a molecule called rubisco, and we'll see what that name stands for. There are some other paths that also get used, the C4 pathway, and we'll see why that's a useful thing. Having fixed the carbon, we're then going to want to rearrange those molecules through a series of reactions called the Calvin cycle, and then that's then going to provide for carbohydrate synthesis. So that's the grand overview of carbon fixation. Is that legible to you up there? Can't tell from my angle whether that's... It's what? It's quite strange because it's quite brilliant in front of me. And it's, well, I'll have to figure out what's going on there. I don't understand. Okay. Rubisco. Rubisco is the shorthand name that biochemists came up with for an enzyme called ribulose bisphosphate carboxylase. So now I know I would call it Rubisco. And its full name tells you what it's going to do. It's a carboxylase, so it's going to add a CO2 to a molecule, ribulose bisphosphate. Rubisco is a really, really stunning enzyme. It is able to add a carbon dioxide for free. And we've seen a couple of reactions up until now where we're adding the carbon dioxide, and it hasn't been free. It's often cost 2 ATPs or something. But this is a very smart enzyme, and it can do it without involving any ATP in the location. In the process of adding that carbon dioxide, it's going to split a 6-carbon sugar into 2 3-carbon fragments. And I'd like to go through and sort of talk a little bit about what's going to go on here, because it's such an extremely good enzyme and turns out to be so important for life on Earth. Rubisco is the reason we're here. Since this is what fixes carbon, without it, we wouldn't be here. It's really very strange. I don't know how to... Well, so your handouts are legible, yes? I'm not quite sure how to figure out how to translate those things. Anyway, here's ribulose bisphosphate. So it's ribulose. Sounds kind of like ribose. Probably a 5-carbon sugar, and it is. 1, 2, 3, 4, 5. Bisphosphate, 1, 5. 1, 5 bisphosphate. So here's ribulose bisphosphate. It's a keto carbon, sort of like fructose. And this is going to be the target of Rubisco. The enzyme is going to be able to pull this proton on carbon 3 off. And there's a magnesium in Rubisco that may play an important role in kind of stabilizing all of that. So it's going to make a structure that looks like this. So it's ene-diolate. So it's if it still had its proton there, it'd be an ene-ol, right? An ene double bond alcohol configuration, which we heard in phosphoenol pyruvate, for instance. But this is an ene-diolate. It's an ion. If that proton was pulled off. So this is now a charged group in there. This ene-diolate, this charged, this deprotonated alcohol, if you like, is able to attack the carbon dioxide. And so the carbon dioxide is going to come in and add itself onto that carbon there. All right? So now we actually have a 6-carbon fragment, still phosphorylated at both ends. Have our proton back there. This is a single bond now. Other intermolecular rearrangements, so we now have a carbonyl carbon there. This looked uncomfortable, doesn't it? It's got these phosphates up there. It's got another negatively charged group there. This just looks like the sort of thing that would like to fall apart if it could. And in fact, the other thing that Rubisco is going to do is use water to attack in between there. The water is going to attack on the C3 and split the molecule like so. And so we now have 2 3-carbon fragments. And if you look at these carefully, you'll see that both of them are 3-phosphoglycerate. So we now have 2 molecules of 3-phosphoglycerate. So a very slick way of producing the products we need. This is the carbon that got fixed, added into the molecule. This was carbon dioxide a second ago. Because in the process of splitting the molecule like this, you've freed up these 2 carboxylic acid groups. See, here's this potential. This is at the edge of being a carboxylic acid. Because you've freed this up, the resonant stabilization of those groups provides some of the energy to go forward into the products. Rubisco is the most abundant protein on Earth. There's more of it than anything else you can think of. And it is the thing that provides the 3-carbon fragments that can be used for all the rest of metabolism. The 3-carbon fragments will end up in sugar, they'll end up in amino acids, they'll end up in lipids, they'll end up in nucleotides. All of them eventually come traceable back to Rubisco. It is not, however, very efficient, or at least very fast. It can catalyze 3 reactions per second. I don't know if you remember when we were talking about enzymes and how fast some of them were, things like triose phosphate isomerase that could literally be as fast as diffusion. That's not Rubisco. If TPI is a Ferrari, Rubisco is roller skates. And it may well be that it's because it's not very fast that there's so much of it. If it could do it any faster, they wouldn't need so much of it. In land plants, it actually is a multimer. This is a space-filling diagram of the structure of Rubisco. But it's not allosteric. It's just a multimer, and one possibility that has been considered is that it has to be a multimer to fit enough into the chloroplasts for things to work. This is an example of Rubisco from bacteria up here. So Rubisco catalyzes a really amazing reaction, but it's not a perfect enzyme. It would be the perfect enzyme if it would just catalyze that reaction and nothing else. Unfortunately, Rubisco is not very specific. And so there's another kind of reactions that can also catalyze that are called photorespiration. Now in us, respiration is when we consume oxygen. So photorespiration is consuming oxygen in the presence of light. And that happens when Rubisco uses oxygen as its substrate instead of carbon dioxide. And what would that look like? Let's start off with a ribulose bisphosphate. That definitely looks better there. So here's ribulose bisphosphate. It's phosphate at each end, a carbonyl oxygen there. But now if we add in an oxygen instead of a carbon dioxide and split it with water, we end up with simply a 2-carbon fragment and a phosphoglycerate. So we have not generated new carbon. This molecule is called phosphoglycolate for historical reasons. Phosphoglycolate is not by itself particularly useful. And to get those carbons back so we don't lose them altogether, we have to actually invest ATP and NADPH to recover it. So photorespiration is a wasteful process. It is not useful. Actually, it costs energy. So it's competing for carbon fixation as a process. I think we'll maybe call it a break there. We're not quite to 4:20, but I think we want to cover the Calvin cycle all as one go. So.